sharing experiences, really important, learning from each other, hearing from our expert speakers.
I want everyone to really take control of their health, because it's your body and your life, and if you're not in control of it, no one else will be.
Turns out that almost all pacemakers are completely fine to go through an MRI scan.
Both septal ablation and surgical treatment show good results. It's safe, comparable.
HRT for many women is a lifesaver.
However, and it was a real shock, they told me I did need a transplant.
It's funny being a carer. You know, sometimes people don't take their medicines properly and you get cross at them. Why are you not taking your medicines?
I would say the next big stage in the development of this story was our understanding that this is, in many people, a genetic condition. So it's a heritable and transmissible disease.
So some of the pros of genetic testing are that we can find a causative gene change in some people.
All right, well, welcome to the session. My name's Betty. I'm a cardiologist and a clinician scientist in Oxford, and I love looking at images of the heart. I do MRI scans of the heart for any of you who've had one, and I use very sophisticated approaches to study the heart in great depth. But what I'm doing today is slightly different, so I'm going to talk to you about the CureHeart initiative, which is a BHF-funded project, and it has some ambitious goals. So you've all heard about hypertrophic cardiomyopathy in previous talks, but I'd just like to show you some images to explain how we make this diagnosis for patients. So on the left is a scan of a healthy person who has no heart muscle problem, and on the right is an MRI scan of a person with hypertrophic cardiomyopathy. And you'll see that this is a transverse or a cross-sectional cut of the body, and the heart sits just behind the chest wall, and it's sort of wrapped around by the lungs and other organs. And there's a black band in the middle of the heart, which is the septum. And in people with hypertrophic cardiomyopathy, this band thickens. So the example on the right, I hope you can appreciate, it's quite a thick muscle in the middle of the heart. But how does this affect the heart? So the heart's quite a sophisticated organ. It's got electrical cables that tell it to beat and pump regularly. And when the muscle gets thickened, these cables can wear out and you can have abnormalities in the conduction. And these conduction changes can cause the heart to stop suddenly, and one can have a cardiac arrest. Alternatively, it can affect the pumping function of the heart, such that the heart is not pumping blood to important vital organs like the brain and other parts of the body. So here's an example of just a movie picture of the heart, and the one on the left you can see fills in quite nicely. There are top chambers that store blood to fill the bottom chamber, which is your pump, and in the left you can see the heart relaxing and contracting quite nicely. On the right, in our patient with hypertrophic cardiomyopathy, the heart is stiff. It doesn't have much room to fill, and even when it does squeeze, there's a bit of reflux of blood into the reservoir, the top chamber. And so this is part of the problem and what contributes to heart failure in people with hypertrophic cardiomyopathy. But are there any other diseases of the heart muscle? Certainly. So the muscle of the heart can be thin and weak, and this is what is referred to as dilated cardiomyopathy. It can be stiff and not relax well, and that's been discussed previously, heart failure or diastolic heart failure. But also another term that's used to describe this is restrictive cardiomyopathy. And because the muscle is so complex, it not just has the pump, but also the electrical aspect. You can have electrical abnormalities which contribute to fatal rhythm disturbances, and that this form of cardiomyopathy is called arrhythmogenic cardiomyopathy. So one of the things with these cardiomyopathies is we know that a proportion of individuals carry genes that cause the condition. We know that it's quite common in some cases, affecting as many as 1 in 250 individuals, which means that there will be people out there in the population who may have the disease but may not have any symptoms. And that includes even young children in schools, as well as people who go to see their GP for something else completely different. They may not have symptoms suggestive of cardiomyopathy. So it is fairly common, and because of the genetic background, it can be inherited in families. And as I said, because it is a disease of the muscle, it can affect the electrical conduction, but also the pumping function, and can be a cause for heart failure and sudden cardiac death. Now, you'll all be familiar with these two images. These are tragic stories of two young athletes that collapsed on the field. So actually, the image on the right are pictures of team players of the Danish football player who collapsed during the World Cup. And basically, this is a story that is all too familiar to us, and it really drills down the importance of understanding what causes these fatal arrhythmias and collapses in young people, and also highlights that there are people out there who collapse and who are not in a visible arena or environment to be saved. So there are events like this that are occurring quite regularly, and people are just not aware of it. So it's really important for us to think of this and how we can find solutions to identifying people early, but also treating them appropriately. So we know that the cardiomyopathies can be caused by genes, but also some environmental factors. In some cardiomyopathies, about 20 to 30% of individuals have a single gene that is responsible for the phenotype or the appearance of their heart. And in others, you'll find that there's a number of common spelling mistakes or errors in their genetic makeup that contributes to the manifestations or the appearance of the heart in these people. So going back to a bit of biology, you've probably seen some of these slides before, but just to revise or go through the genetic etiology. So the heart's made up of cells, which are little compartments, complex compartments that have machinery that contract, and that requires energy and depends on the mitochondria, which is a powerhouse within these cells to provide energy. But at the core, fundamentally, these cells depend on a molecular motor. And what we know now, thanks to research, extensive research into the genetic origins of some of our cardiomyopathies, is that genes that code for proteins that are important for this motor can be affected in the cardiomyopathy. And what are genes? So genes are nothing else but an information leaflet for our body to develop and form organs. And just like in an information leaflet, you have words and letters. The genes can have spelling mistakes in these information leaflet that can cause the heart to form abnormal proteins, or any changes in the heart that may contribute to heart failure or electrical abnormalities. So here's an example where there has been a change or a spelling mistake where one of the nucleotides or fundamental building blocks has been altered, and what this has resulted in is a faulty protein. Now, if this faulty protein is really important, it can cause a malfunctioning motorhead, and this can result in the disease, or it can cause a disease in a person. So even one abnormal spelling mistake or one abnormal nucleotide can contribute to abnormalities in the motor heads and can cause the disease, and that's called dominant negative genes. And how do you fix this? So one can develop treatments to silence or correct this faulty gene, and that should hopefully prevent someone from developing the disease. Another spelling mistake that can cause cardiomyopathy is something called haploinsufficiency, where a spelling mistake causes the formation of that protein to stop, or there's less protein being produced as a result of that spelling mistake. And what happens as a result of that is you don't have sufficient proteins and the motor is just malfunctional. And so in this case, how you treat this is by increasing the production of the copy of the gene. So this is one way by which you can target the gene to fix the disease. Now, in terms of silencing the gene, there are a number of ways that this can be done. So I'll just briefly talk about two approaches. One is where there's molecules of RNA that essentially can be given to the person and interacts with the gene and stops it from expressing, or stops its expression in the person. Another way is using a very clever technology called CRISPR-Cas9. It's a molecular scissor that goes directly to the faulty gene and essentially makes a cut, snips off the abnormal gene, and lets the body repair itself. And this is actually a creation of God, in the sense that it's a biological process that exists in nature. It was originally described in bacteria, and this was a defense mechanism for bacteria to kill viruses that infect the bacteria. And so we can use this technology to essentially fix faulty genes. Another approach, which is something that was developed by a very clever scientist and his group in the United States, is base editing, where you can essentially send a chaperone, or again, like CRISPR, a genetic vehicle that goes in and essentially just fixes or corrects the spelling mistake. So it doesn't make any cuts, but just edits the gene and fixes the problem. But it took years to get to this point, years of research for us to be at this place where I talk to you about some clever techniques and how to fix and edit the genes. And what we know is there have been numerous preclinical studies where it's shown to not successfully reverse the disease, and also essentially behave like a cure for people with genetic heart diseases. However, when it comes to introducing this in humans, there are a number of challenges with these treatments. And this is where CureHeart and the investigators of CureHeart are working towards finding solutions. So the first is that these treatments need to be really precise on what they target, because as I showed you, it could be one letter in the genetic information leaflet that's abnormal. So not only does the vehicle have to get into the heart, but has to get into the cells and correct that part of the genetic makeup to fix the problem. The second is the delivery of these genetic treatments, genetic cures, is complicated because the body's got a very powerful immune system, which treats anything that's external as an invader, and so it fights it off. So the efficacy of these treatments can be muted as a result of our body's immune system. The other thing is, because all this is fairly new, we don't quite know what the long-term effects of these treatments are in people. And of course, you've heard that some of these therapies are extremely expensive. They're really costly. So how do we make this accessible to the wider community? And finally, just because you carry a gene that causes the cardiomyopathy, it does not mean that you will develop the disease. So there is something called variable penetrance, which means that some people may develop the disease in their lifetime and others don't. And so we need to be able to identify those who are at risk and target them early before they develop the disease. At present, the thinking is that if you deliver these treatments too late, once someone develops the disease, it may not be effective in its ability to prevent or cure the disease. And so one of my work in this group is to use imaging approaches to identify people who are at risk of progressing and developing the disease. And for this, I use sophisticated technology like MRI and really advanced approaches on MRI that does not require radiation or any other thing that might be, any other exposure that might be toxic to our patients, and that allows us to really provide detailed tissue-level insights in the human hearts of people who carry the gene that causes the cardiomyopathy. So I'd just like to wrap up by saying that the CureHeart's ambition is impressive, and it has some really immense potential. But of course, there are some potential challenges that we need to overcome. And If we are successful in our endeavors, I'm hopeful that this will set the stage and pave the way for genetic therapies in other cardiovascular diseases. And to do this, we could not have come to this point without the support of the key investigators. So the real brains behind this work are Professor Hugh Watkins, Christine Seidman, and all the other members on this panel who had this vision that it is possible to develop treatments that might potentially cure patients at risk of developing cardiomyopathies. So with that, I'd like to thank Cardiomyopathy UK and all of you for your attention and welcome any questions at the end.